rapid fire review. We'll see how it goes. I would love feedback on it. So I'm going to try to do this every PowerPoint that we went over the whole year, and we'll see how it goes. So you should know about atoms and elements, in particular protons, neutrons, electrons. All atoms are electrically neutral. Remember, the atomic number equals the number of protons and the number of electrons. So if you look at a periodic table, this is the atomic weight, protons plus neutrons. This is the atomic number, number of protons, and number of electrons. Isotopes are atoms with the same element with different numbers of neutrons, and they're often unstable. And that instability can lead to radioactivity, meaning that they spontaneously break apart, and that often happens, again, in different isotopes. Electrons form in shells, and the most important thing for you to remember is that the outer shell is the most important for if they're reactive or not. If they're not reactive, that means they have a full outer shell of electrons. If it's not full, then they want to react with other atoms to make sure that they are full. They like to fill in these outer electron shell vacancies, right? There are multiple kinds of bonds that can hold things together. Ionic, meaning that ions, positive and negatively charged ions, are interacting. Covalent bonds, meaning that they are sharing of electrons. And hydrogen bonding, between polar molecules, right? Not between, they're between molecules. This chart, I think, is really nice. Remember that polar bonds come in two flavors, nonpolar and polar. So ionic bonding is when, here I'm going to go ahead and play that, sorry. Ionic bonding is when one atom loses an electron and one gains it, and those ions come together. And here's some practice questions if you want them. Covalent bonding is sharing of electrons in that outer shell, all right? Sharing. So that happens when we form water, for example. We are sharing electrons. Hydrogen is now happy, and oxygen is now happy. Looking at just these, you should be able to figure out how many covalent bonds are formed most often with these molecules, right? Covalent bonds, again, produce either polar or non-polar molecules. Polarity is a huge thing in biology, so you need to know that, right? So, for example, these have partial negative and partial positive charges because they are unequal in their electronegativity, all right? If you have same atoms, or if you have CHs, those are non-polar covalent bonds. They have the same electronegativity. So non-polar, they're the same. Polar, different, like polar opposites. Water is probably the most important polar molecule for biology. Again, oxygen is partially negative. It's pulling those electrons towards it. Hydrogens are both partial positive. Hydrogen bonds occur between polar molecules. Water is the best example of hydrogen bonding, right? So hydrogen bonding are dictated by these dashed lines between partial negative and partial positive, between separate molecules, right? Some of the other molecules that can be involved in hydrogen bonding, H's and O's are the most common, H's and N's, and H's and F, which don't occur much in biology. Hydrogen bonding is comparatively very weak, but collectively very strong. Covalent bonding is the strongest. They're sharing electrons. Hydrogen bonding, a single one is very, very weak. Collectively very strong. We see this with holding together the two strands of DNA. Hydrogen bonding is not only between O's and H's, but you can see it here between N's and H's when we have the base pairs coming together. We have some practice questions. You should be able to pick out, for example, polar parts here and here, and non-polar parts here. Water is super important for life because it has cohesion, which leads to surface tension. It is a very good solvent, interacts with many molecules, and it has a very high specific heat. It moderates temperature change. So again, water is very important, as we saw, remember the Jesus Christ lizard, and water going up a tree in transpiration. Cohesion, like molecules attracting, adhesion, unlike molecules. So when we have transpiration, we have like and we have cohesion among the water to itself. We have adhesion from the water to those walls, right? Water is a very important solvent. It dissolves a lot of good things. And we always saw that if you put a solute in a solvent, it will go from an area of high concentration to low. Remember that the water is also going from a high concentration to low. Water-soluble molecules are hydrophilic, meaning they are water-loving. And that means that they are either ions or other polar molecules, such as sugars and amino acids. Important to remember, though, that sugars and amino acids themselves are held together by covalent bonds, meaning they will break down to individual sugars and individual amino acids, but those things will not break down themselves. Remember that water and oil do not mix, meaning hydrophobic and hydrophilic things do not mix. Water has a very high specific heat capacity, meaning it takes a lot of energy to change the temperature of water, either heating or cooling, right? A lot of energy to heat water, a lot of energy for water to change into, for it to evaporate. Remember that ice is very, very important. It's an unusual solid. Most substances become denser when they solidify. Water is different. When water forms ice, it forms these very nice lattice where the water molecules are separated from each other in this very crystalline structure, making it less dense. There's more air in there, right? So water, very compact. Ice kind of spread out, making it less dense, meaning fish can live in there and they don't die in the winters. Remember about acids and bases, how water can break apart into hydroxide and hydrogen ions. Acids have a low pH with a lot of H's. Bases are a very high pH with a lot of OH or hydroxide ions. Neutral is about pH 7. Mr. Anderson has a good review if you need a review on that. Buffers help maintain a constant pH. And this is very, very important in our blood system and for any protein that you're dealing with. So if you're studying a protein in lab, you need to have it in a buffer so that its pH does not change, which would cause a denaturation. All right. And I'm going to switch to the next one now. All right, so biological molecules. This is a little bit of a review. Carbon is the most important biological molecule because it can form lots of different bonds, all right? So it can form 4 different covalent bonds with different things. So whereas hydrogen can only form 1, carbon can form 4, meaning it can have lots of different structures, leading to very complex shapes, branches, rings, sheets, and helices. Remember, if it's organic, that refers to having hydrogen bound to oxygen molecules. I think I said, hydrogen, I said oxygen, sorry. Hydrogen bound to carbon molecules. Life completely depends on carbon and its ability to form these 4 different covalent bonds. Carbon skeleton, chain of carbon atoms, lots of different structures can form. Remember your functional groups, all right? So we have 7 of them: hydroxyl, methyl, carbonyl, carbonyl, carboxyl, amino, phosphate, and sulfhydryl. You should know these and be able to point them out on a diagram. You should also know whether they are polar, if they form hydrogen bonds, etc. Right? The two kind of weird ones are these two in terms of their polarity. Methyl is nonpolar, as is sulfhydryl. The rest have are polar molecules, and that has to do with their electronegativities. If you look at a single molecule, you should basically be able to tell me something about it. This is polar or nonpolar. It actually has two parts. This part is nonpolar, this is polar, meaning it is amphipathic, right? When we talk about biological molecules, they all have monomers that come together to form polymers through a reaction called dehydration synthesis, where we are putting two things together to remove a water. When we, to form dehydration synthesis. When you are cutting something, it's called hydrolysis. You add water into the molecule to break things. That's called hydrolysis or hydrolysis. So there are four types of biological molecules that you need to know. They are polysaccharides, meaning carbohydrates, fats, lipids, proteins, and nucleic acids, right? Carbohydrates made of CHs and O in this ratio 1 to 2 to 1. The monomers are monosaccharides. They come together to form disaccharides or polysaccharides. The most common carbohydrate is glucose. Polysaccharides are very important for storage. Starch is for the high energy storage in plants. Remember, glycogen is what we have for animals. We also have structural polysaccharides in the form of cellulose, which we cannot break down, and chitin, found in exoskeletons and in the cell walls of fungi, right? Lipids are hydrophobic molecules. They're the ones that are hydrophobic. They're composed almost entirely of carbon and hydrogen, right? They're hydrophobic. They have a number of different functions, and we will go over all of them, but in particular, there's oils, fats, and waxes, phospholipids, which make up biological molecules, and steroids, which form those carbon rings, which are involved in cell signaling. So here's your fatty acids. Remember, big long chain with a carboxyl group on the end, carboxylic acid. They're primarily nonpolar. Yes, a little polar, primarily nonpolar. Fats, 1 glycerol, 3 fatty acids. You can look at the structures here. Wax, 1 alcohol, 1 fatty acid. And then we have saturated and unsaturated kind of fats here, where saturated, meaning that they are solid at room temperature. Unsaturated, there are double bonds making it kinky. Remember, oil is kinky. Phospholipids, main component of cell membranes, right? We have a glycerol group, we have 2 fatty acid chains, we have a phosphate group, right? These are amphipathic, polar head, 2 nonpolar tails. You should know the basic kind of structure of this. You should be able to interpret data here if given these types of things. Steroids, nonpolar, they go straight through the plasma membrane. They are small and nonpolar. They can slip right through. They are all based on the structure of cholesterol, usually modified from that. Proteins are formed of amino acids, and they come together through peptide bonds. They are the most diverse in terms of structure and function, but they're all based on 20 amino acids, right? They form enzymes, which are very important for making reactions happen. Here are some examples of structures and functions, very diverse. This is 2 amino acids coming together to form a dipeptide through a peptide bond. Right? Multiple amino acids together form a polypeptide or a protein, a chain of amino acids. Proteins can be denatured depending on what environment they are in. Their folded structure depends highly on different types of bonds, which can break in different, if the pH is different, or if the temperature is different, or if the salinity, etc. So you should be able to tell this is polar, polar, nonpolar, 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 right? Hydrophilic, hydrophobic. Don't forget that cysteine is that one special amino acid that can form disulfide bonds. Proteins have 4 levels of structure. Primary structure, which is just the sequence of amino acids. Secondary structure, held together by hydrogen bonds, alpha helices, beta pleated sheet. Tertiary structure, complex folding, held by interactions of R groups, right? Hydrogen bonding, ionic bonding, polar binding, etc. Quaternary structure, 2 chains coming together. So primary, secondary, again, these are hydrogen bonds of the backbone. Tertiary, held together by R groups. In particular, these can be disulfide bridges as well in the tertiary and quaternary structure. Function is directly dictated by structure of proteins. Nucleotides are the monomers of nucleic acids. The 2 most important ones are DNA and RNA. All nucleotides have 3 parts, a phosphate group, 5-carbon sugar, and a nitrogenous base. And that can be A, T, C, or G for those bases. ATP is an example of a nucleotide. Here we have 3 phosphate groups instead of only 1. The big difference between DNA and RNA has to do with the sugar involved. Ribose is deoxyribose has a lack of an oxygen on that 2' carbon, whereas ribose has an OH. These are molecules of heredity, and remember, phosphate backbone held together by phosphodiester bonds. Those are highly negative, making the DNA molecule negative, held together by these hydrogen bonds of the bases. Remember, pure as gold for adenine and guanine for the purines, or cut, C-U-T, the pi for pyrimidines. And only when pyrimidines and purines come together do you maintain the kind of thickness of your DNA molecule, right? Remember, DNA structure is an anti-parallel, meaning 5' end is at one side, and that represents a 5' phosphate. On the other end, it is the 3' end, which is the 3' carbon sitting out there, all right? And that is where we will stop the first video.